we did DNA replication, which is the process by which DNA is copied in the cell before mitosis, meiosis, and of course binary fission. And here you can see our original DNA strand, which is in a double helix, and it's made up of nucleotides, which you can see here, which are thymine and adenine, which pair together, then cytosine and guanine, which also pair together. And
they are together by hydrogen bonds. And our cake displays how the nucleotides separate along the strands, and it serves as a template for new complementary strands, and they move to new cells during cell division. Okay, so first you can see that the enzymes are separated by, this is the helicase, this giant purple blob. And it separates or unwinds the DNA strand, and by breaking the hydrogen bonds, which are the, I don't know if you can see them, like the metal. Between the complementary nitrogenous bases, which are, as she said, those. Okay, so then you explain more.
Yeah.
Where is it?
Okay, so over here you can see coming out of the helicase, you can see a wide sort of shape, which is known as a replication fork. And as this fork moves along the original DNA, the synthesis for the strand moves in the opposite direction, so you can see it kind of like curving around this way, because here it goes from 5' to 3', which is the way that it moves, whereas on this strand it's going from 3' to 5', so it needs to pull it around and like reverse it. And then this creates some little spaces in the DNA, which are later brought together with DNA ligase. And then Kenny can.
So these chunks of sugary goodness, they're called DNA polymers. So they add complementary nucleotides, which can be found like floating around through the cell. So, so here you have like an adenine, so you add a thymine, and then here you have a cytosine. You can add a guanine.
And as they're added, the covalent bonds form between the deoxyribose sugar and of one and the phosphate group of another.
And with that bond, there's also hydrogen bonds being formed between the two nitrogenous bases. For example, with this one, they haven't quite formed yet, but on the next pair, they have. Then hydrogen bonds, as I said, form between these and the original strand, which you can see here, and then the new strand. And this is called semi-conservative replication, because one of the original strands is kept, whereas a completely new one is brought in on the other side.
So after the DNA polymerase adds in complementary nucleotides, they fall off, and then these strands are ready to move for the new cell for cell division.
Which includes mitosis, meiosis, and binary fission. The end.